These are the recorded lectures for Chapter 10 on meiosis and sexual reproduction. There are a lot of sections in Chapter 10. The first one is just an overview of meiosis, and then you move down to Section 3, which goes through the individual phases of meiosis, which are very similar to mitosis. There's just two sets. So like mitosis was prophase, metaphase, anaphase, telophase. Meiosis has prophase 1, metaphase 1, anaphase 1, telophase 1, and then prophase 2, metaphase 2, anaphase 2, telophase 2. So there's at least a lot of similarities. So I know I'm telling you this because I know it looks like a lot of information, and it is, but since we've already covered mitosis, meiosis will be a little bit easier to understand. We will talk about other topics besides meiosis because meiosis helps contribute to genetic variation. So there is a section that explains that. And then we'll compare both processes, meiosis and mitosis, and we'll talk about how meiosis falls into the cycle of life, especially when it comes to humans, the human life cycle. And then we'll talk about some chromosomal abnormalities. So, starting with the importance of meiosis, for one thing, meiosis is, the process of meiosis is what leads to a large amount of genetic diversity. You can see highlighted there are more than 70 trillion different genetic combinations possible from the mating of two individuals. More than 70 trillion different genetic combinations when two individuals mate and produce an offspring. And then there are differences in the way that, since meiosis is the process that occurs during the formation of sperm and egg, there's a difference in sperm production and egg production. In males, sperm production begins at puberty, but in the female, egg production starts before birth and ends at menopause. So it starts earlier and then actually ends at menopause, okay? Whereas men can continue to produce sperm for longer than that. Well, the men don't really have a menopause. So the first section of Chapter 10 is on an overview of meiosis. Meiosis is only used for sexual reproduction. You do have some similarities as far as what happens before meiosis during interphase. The same exact things happen. Chromosomes are going to be replicated or duplicated during the S stage of interphase. And then during meiosis, the chromosome number is going to be divided in half, whereas in mitosis, the chromosome number stayed the same. So the parent cells are diploid. They have 2 copies of each chromosome. And then after meiosis, the cells that are produced are called gametes. Eggs and sperm are called gametes, and they're going to be haploid. They're only going to contain, in humans, they're only going to contain 23 chromosomes instead of 46. And then down here it says gametes fuse in fertilization to form a diploid zygote. So when the sperm and the egg unite, the sperm and the egg are both haploid. They both have 23 chromosomes each. When they unite and form a zygote, the zygote's going to be diploid, and it will have 46 chromosomes. Okay, so when we talk about this term homologous pairs, this is going to be new. We didn't use that terminology in Chapter 9 in mitosis. Or we may have, but it's not really necessary to talk about pairs of chromosomes in mitosis. But in meiosis it is, because humans have 23 homologous pairs of chromosomes, and in the sperm and the egg that are produced after meiosis, there's only one one copy from each homologous pair. Okay, so diploid or two-end cells have 2 chromosomes of each type. Chromosomes of the same type are said to be homologous chromosomes. So what it means to be a homologous chromosome means that the homologous chromosome pairs will have the same length. They will have centromeres. Remember, the centromere is where the sister chromatids are attached. So their centromeres are positioned in the same place. One of the homologous chromosomes will have come from the father and one will have come from the mother. The chromosomes also show similar banding patterns because they have genes for the same traits in the same locations. That doesn't mean the genes for those traits are the same genes, same versions of the genes, but they're genes for the same traits. So maybe the gene for eye color or the gene for hair color, they don't have to be the same colors, but they have to be genes for the same traits. And that's what those bands show. So like, okay, let me find a good example here. We'll just look at pair number 1 because you can see them easier. You see the top portion of each of these homologous pairs of chromosomes is green. So that green band represents a particular gene, a gene for a certain trait. And that's what the banding patterns represent. So this is a karyotype. This is a picture of the homologous pairs, all 23 pairs for a human. And this actually happens to be a female because both the chromosomes of the 23rd pair, which are your sex chromosomes, both of them are X chromosomes. So you can see that each pair of chromosomes, each chromosome has 2 sister chromatids attached at a centromere. The centromere is located in the same place. The homologous chromosome pairs are the same length. Their banding patterns are the same. Under the microscope, they look exactly the same. They're going to have different versions of the genes that they code for, but you wouldn't be able to tell that just by looking under the microscope. So this is a picture of all 23 pairs of an individual female human. And that means that for every one of the pairs, for pair number 1, pair number 2, all the way to pair number 23, one member of each pair came from the father and one came from the mother. Okay, now meiosis is a reduction division. In other words, it's a cell division that occurs twice. There are 2 divisions rather than just 1. In mitosis, the cell only divided one time, but in meiosis, the original parent cell will divide twice. And it reduces the chromosome number. So this is an overview of what happens in the first division called meiosis 1, when the first parent cell divides. Before meiosis 1 ever happens, there is an interphase. And during that interphase, the chromosomes are replicated and they contain 2 identical sister chromatids. So in a human, you would have 46 chromosomes, which would be 23 pairs of chromosomes. And all of those would be replicated, so they would contain sister chromatids. Now, during the first phase of meiosis 1, it's called prophase 1, those homologous chromosomes will match up with each other. They will pair up, just like you see in the karyotype here. The homologous pairs will line up side by side. And that is called synapsis. And during that synapsis event, the chromosomes can exchange pieces. They can exchange genes. So there's even more diversity than you would get just by the random pairing up of chromosomes. All right, during metaphase, the homologous pairs line up rather than in mitosis, you would just have individual chromosomes lined up at the metaphase plate. Now they're going to be lined up in pairs, 2 by 2. And then in anaphase, the two members of the homologous pair separate. So in mitosis, sister chromatids separated, but in meiosis, homologous pairs will separate. It's real important that you pay attention to the differences between mitosis and meiosis. That's where a lot of your questions are going to come from. All right, so the last part here is that at the end of telophase and cytokinesis, each daughter cell receives one duplicated chromosome from each pair. And the chromosome number now is reduced from 2n to n. So if we started out with 46, each daughter cell now is going to have only 23 chromosomes. In meiosis 2, I always remember and always tell students that meiosis 2 is the same as mitosis. So really, the only thing you're having to learn that's new is meiosis 1. All you have to know for meiosis 2 is that mitosis occurs for both the daughter cells that were produced in meiosis 1. So meiosis 2 is identical to mitosis. We don't have another interphase. We have a phase called interkinesis, but DNA is not replicated during that phase. It's just a very brief kind of a pause, you know. So after meiosis 1, you don't have another DNA replication. But once meiosis 2 starts, the sister chromatids are going to line up single file at the metaphase plate. Then the sister chromatids are going to separate and move to opposite poles like they did in mitosis, rather than homologous pairs, because there's no more pairs in meiosis 2. The pairs are gone. You just have one member of each pair. So now in anaphase, we have sister chromatids separating, and then we end up with 2 cells dividing, so that gives us 4 cells that still have the haploid number of chromosomes. So all 4 cells will end up with 23 chromosomes. They'll just be single-stranded chromosomes. All right, and this is what the cell looks like before meiosis begins. We have 4 total chromosomes, and you can see the short red one and the short blue one, they would be members of a pair, and then the long blue and the long red would also be members of a homologous pair. So the diploid number right now is 4. Now what happens during meiosis is, first of all, there is an interphase before meiosis, and you can see that those single-stranded chromosomes have duplicated. And what we're looking at is that the homologous pairs have matched up. They're now lined up at the metaphase plate. So this phase here would be metaphase 1. And then the pairs are going to separate, and what you end up, at the end of meiosis 1, you end up with 2 daughter cells that have the haploid number of chromosomes. But both of those chromosomes are still duplicated. They still have sister chromatids attached at a centromere. So now we're going to have meiosis 2. Our 2 cells that were formed during meiosis 1, they're going to divide, and we'll end up with 4 haploid cells at the end of meiosis. Section 2 talks about how the different ways that we get genetic variation from meiosis. So there's 2 key ways that we get genetic variation. There's crossing over, and that happens during prophase 1. And then there's independent assortment, and that's just deals with the way that the chromosomes pair up and line up in metaphase 1. The way they pair up in prophase 1, and then the way they line up in metaphase 1. So the definition of crossing over is the exchange of genetic material between non-sister chromatids during meiosis 1. So what does that mean, non-sister chromatids? When you look at these 2 pairs of chromosomes, one is red and one is blue, notice how this, I wish they would have shown before. The before would have been that this blue piece would have all been 1, okay, just like it is here. You'd have 2 copies that look just like strand number 4. But what happens is strand number 2, which belongs to the red chromosome, and strand number 3, which belongs to the blue chromosome, that's what makes them non-sister chromosomes. They cross over and exchange pieces.